Today we will continue our discussion about this translocation of calreticulum under oxidative stress. In the previous video we have discussed that Perk signaling pathways might play a role towards translocating calreticulum from endoplasmic reticulum to the cell surface. In this video they validated that hypothesis, or we will discuss what are the experiment author has done to validate the hypothesis that Perk signaling pathways might play a role towards translocating calreticulum from the endoplasmic reticulum to the cell surface. Validate this hypothesis, the first experiment they did is to use this Perk siRNA or small interfering RNA. It will disrupt this function of this siRNA or small interfering RNAs is to disrupt the production of the protein against which this siRNA has been designed. So they have used Perk siRNA, and for control they use this control siRNA. It will not disrupt the translation of this Perk protein. So in the control case they have considered it as a 1, and when they use Perk signaling Perk siRNA and there is no oxidative stress, they do not see any difference between the calreticulum mRNA productions, as normal calreticulum mRNA productions does not depend on this Perk signaling pathways. However, when the H2O2 is introduced or oxidative stress is introduced and there is no Perk siRNA, they can see that there is an increase in this calreticulum mRNA, and this increase is significant compared to the control case. And when they use this Perk siRNA along with the oxidative stress, they see the calreticulum mRNAs goes down, and those difference is statistically significant. So this indicates that Perk signaling pathways might play a role towards upregulation of this mRNA of calreticulum. And after validating that the Perk signaling pathways has a direct relationship with this calreticulum mRNA, they check these PIG1 cell lines in the immunostaining, and they can see that when they use the control siRNA with oxidative stress, the calreticulum signals are more prominent on the membrane of the cells. However, when they use this Perk siRNA, those signals goes down, and they do not see more calreticulums on the membrane of the cells. And these things they have quantified on the right hand side, where they can see that decrease in the calreticulum's fluorescence intensity is statistically lower compared to the case where control siRNA is used. And after confirming that Perk siRNAs reduce the calreticulum translocation on the cell surface, they again validated their results using flow cytometer at different time intervals of the oxidative stress, and in this case they use this Perk siRNA. So for the control case, they can see the highest signals of the anti-calreticulum. However, when they however with time they can see that reduction of this calreticulum signals on the cell surface, and those differences are statistically significant compared to the control case. So this indicates that the Perk signaling, with the disruption in this Perk signaling pathways, the calreticulum's translocation to the cell membrane will also get disrupted, indicating that Perk signaling pathways plays a role towards translocating calreticulum from the endoplasmic reticulum to the cell surface. And these things are also in line with this Western blot result, where they can see that the signals for the calreticulum goes down compared to the control case in the membrane section, in the membrane fractions. However, for the cytoplasm the signals are almost same, and the quantification of this Western blots is shown on the right hand side, where we can see that a decrease in this intensity of the calreticulum is statistically significant in the membrane case. However, the difference is almost, there is no difference, no statistically significant difference in the cytoplasmic fractions. And after validating the results on the cell lines, this PIG1 cell lines, they decided to check their hypothesis on the patient's skin. So they used two different patient skins. First one is healthy skin and another one is vitiligo PL skin, and they check the perk signals, and they can see that healthy skins has lower perk signals compared to the case of this vitiligo PL skin. That is, the green signals are more prominent in the case of vitiligo PL skin, indicating that the perk signaling pathways are upregulated in the vitiligo skin patients, which and since they are also under oxidative stress, so they will have more calreticulums that are translocated on the cell surface. So it might be uh, one of the important functions that might occur during this vitiligo skin disease. And the quantification of this immunofluorescence intensity is shown on the right hand side, where they can see that fluorescence intensity of the perk is statistically significant in the case of this vitiligo PL skin compared to the healthy skins. So from this figure, or from the results that are presented in this figure, they validated their hypothesis that perk signaling pathway helps in the calreticulum translocation on the cell surface and oxidative stress in the melanocyte cells, since they have used these PIG1 cell lines, which is the immortal cell lines derived from melanocyte cells, or the skin cells. If you need to understand more about their experimental details or the experimental methods, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.